Hello, this is Dr. Clark, and today we're going to go over blood, lymph, and immunity. And so, first off, we're going to start off with talking about blood and the functions of blood. So, blood has multiple functions, and one of the main things it does is transportation. So, it's going to take oxygen to the cells, it's going to carry carbon dioxide away, it's going to take nutrients like glucose, amino acids, different proteins, lipids. To the side of the cells, and it's going to take those waste products like lactic acid and stuff away from there. It plays a role also in carrying hormones and different chemical messengers throughout the body, and that's a very important role that it does to itself. Also, with regulation, it also helps to keep body temperature even through circulation, so it actually helps to warm like the digits and the distal limbs, and then actually helps to actually cool the internal organs. It keeps a constant amount of fluid content to the tissues, and it's very important in keeping acid-base balance in check throughout the body. So it's very important in playing a role with pH. It's also a very important part of the defense system for the body with the immune system because it's where most of the white blood cells are going to be transported to throughout the body as they hitch a ride through the blood supply, then they get off where they need to go to. A couple other things to know about blood is what it's made up of is it's going to be made up of red blood cells, white blood cells, and then you have plasma. And so plasma is going to be a mix of those proteins, electrolytes, hormones, all the other things. So all the liquid fraction of blood is pretty much going to be that known as serum or known as plasma that's going to circulate throughout the body there. So composition right here, when we're looking at it, is this liquid portion here is known as plasma. And then you have the cellular portion, which is your red blood cells, white blood cells, and platelets. And depending on which species we look at is what the percentage is. Right here, we're probably looking more at a dog, which is about 45% red blood cells. So they're somewhere between 45 and say 55% red blood cells and between 45 to 55% plasma. Other species like a horse is more like 28% to about 72%, 28% red blood cells to 72% plasma, and they can go up to about 45 to 55 like this split. So there is kind of a difference between species as far as that goes. When we look at the breakdown of the plasma, about 7 to 8% of plasma is made up of protein. Greater than 90% of water is water and dissolves electrolytes in the water, and then the other 2% are other solutes. So you're going to have different nutrients, waste, gas, and then hormones are going to make up that other part. When we start looking overall across mammals at the fraction of the cellular portion, usually about 2 to 5,000 platelets are found per milliliter of blood. You're going to have 6 to 17,000 leukocytes, just depending on the species, what that range is going to be, and somewhere between 5.5 and 8.5 million erythrocytes per milliliter of blood. And so when we're looking at this, we're all looking at about a milliliter of blood right there. So plasma makes up anywhere from 45 to 78% of the full blood volume, just depending on the species of the animal you will look at. So a lot of times your larger animals will be a little bit more hemodilute. Some of your small animals are going to be more hemoconcentrated, meaning hemodilute, meaning more plasma, less blood cells, hemoconcentrated, meaning more red blood cells, less plasma. And plasma itself is the majority of it ends up being water. And then there's a bunch of things that are going to be dissolved and suspended. So some of the things are like your proteins in here. And those are going to be albumin. And so albumin is the main protein. It's very important for what we call oncotic pressure. Oncotic pressure actually keeps fluid inside the bloodstream and doesn't just let everything go out into the extracellular space and that interstitial space around cells and just cause swelling everywhere. So very important for that. Globulins, these are immunoglobulins, very important for defense. And then fibrinogen is a protein that's very important for blood clotting. We have our gases, there's oxygen, carbon dioxide, and then there's actually even nitrogen in our bloodstream. And part of that's because the majority of the air around us is actually made up of nitrogen. And so, of course, it would go to stand that as we breathe in and out, we're going to have some nitrogen in our bloodstream since it makes up somewhere around 70% of the air around us ends up being nitrogen. And then we have different nutrients like lipids, amino acids. You have metabolic waste, and of course all the different electrolytes with sodium, potassium, chloride, all sitting there, and calcium all inside of the bloodstream in the plasma. Cellular components, when we start looking at those, we're going to have erythrocytes. Their main job is to sit there and carry oxygen. We have thrombocytes, which are going to actually sit there and stop bleeding out of the vessels. If there's any type of damage to the vessels, it actually stops any leakage. And then we have our leukocytes. And when we look at leukocytes, we have what we call granulocytic and agranulocytic. And these perform functions within the immune system. And so it can be attacking bacteria, it can be making antibiotics, it can be, or antibodies, I should say, not antibiotics, but antibodies. Um, they can also be very important in inflammation, calling other cells and other things to an area of local injury. They clean up tissue, they kill cancer cells. So leukocytes have many, many different roles they do, and these are just some of the different cells you can see over here. Erythrocytes in mammals, these are going to be anucleated. They actually lose their nucleus. They're going to be biconcave. They're fairly flexible. They're made this way in mammals because it makes it very easy for them to fit through the capillaries. And this is kind of a good representation and size of them. Over here you can see how small platelets are. Platelets are actually not cells, but they're pieces of cells. They actually break off of something called a megakaryocyte. And so these little pieces that are pinched off go out there, and they're very important for helping to form blood clots. These right here are granulated white blood cells. So you have your basophil, your neutrophil, your eosinophil, and then you have your agranulars, which are going to be things like your lymphocytes and your monocytes. Hematopoiesis, this is actually sitting there making blood cells, and so all blood cells arise from red bone marrow, and they're called from pluripotent stem cells. So these stem cells sit there and they're able to determine and make multiple things. They might make a megakaryocyte that's going to turn out and churn out a bunch of platelets if platelets are low. If red blood cells are low, all at once they'll turn and they'll start making red blood cell production. If white blood cells are low and there's an immune response, all at once they can convert themselves over to making white blood cells. So the stem cells can change their purpose as to what the body actually needs. Fetal hemopoiesis ends up occurring in the liver and the spleen, and then neonatal, sometime after you're born, there's still the liver and spleen involved, but as a neonate, they become less and less involved, and the bone marrow takes over more and more of the responsibilities of making blood cells in the red bone marrow. In older animals, as we actually grow and we get the number of our red blood cells up, and we don't have to produce as many because we've produced so much. So as you're growing, when you were younger, the more you grew, the more red blood cells you need. So you kept a lot of red bone marrow trying to keep up with that growth. Well, once your growth levels out, you just have to try to maintain the red blood cells. And so a lot of your red bone marrow that was throughout the long bones ends up actually converting to yellow marrow, which is fat. And so that red marrow is usually just found at the ends of the long bones and in your ribs. And the thing about it is that if you ever need it to start producing a lot of red blood cells again, if you became anemic for some reason from blood loss or change in diet or something, and all at once you need to start making red blood cells, that yellow marrow can actually, that has fat, can actually very easily convert back into red marrow. There is some capacity, limited capacity from the liver and spleen to actually have a role in hemopoiesis or making red blood cells and some white blood cells, but that's only in very severe anemia, and it's a very limited amount that they actually can contribute to it. So those hemopoietic cells, I already told you, they're pluripotent. They can develop into any one of those bloodlines at any time, and it just depends on which hormones are going to be there to stimulate them. Production of red blood cells, there's a hormone that comes from the kidneys called erythropoietin. Erythropoietin is actually triggered by low oxygen tension to the kidneys. So if its kidneys are not getting enough oxygen, they actually release this hormone called erythropoietin, and it actually goes to the bone marrow and says, hey, we need more red blood cells. We're not getting enough oxygen. Go make more red blood cells. And so this is what it actually, the hormone that actually triggers the production of red blood cells. Animals in kidney failure, a lot of times we do have problems with them not producing enough erythropoietin. There's actually a synthetic form. They use it in people. We can actually use it in animals, and we've used it in dogs and cats to trigger red blood cell production. The only thing is it's actually quite expensive to use. There's a lot of multiple steps in maturation of red blood cells. So they start off nucleated, and then over time, they start to flatten out. They start to actually lose their nucleus until they eventually, when they mature, they're anucleated and they're that biconcave shape. That biconcave shape does a couple of things. One thing is it actually lets the cell pack in more hemoglobin by losing the nucleus and being that shape. Makes it a lot easier and more efficient for oxygen change. And then, like I said, it makes it a lot easier to go through the capillaries. It can easily go through very small, thin capillaries, and they're quite pliable, red blood cells are. Red blood cells themselves are made up of about 65% water, 35% solids. Primary solid is hemoglobin. So they're round, anuclear biconcave discs in mammals. There is a variability in size between species of mammals. And they use plasma glucose as their primary energy source. So that's very important to know. So if you leave a blood sample sitting out too long to wait to do a glucose, you will get an abnormal reading or a low reading from it, because they actually will sit there and start using that glucose and the oxygen within inside of that blood to actually keep themselves alive. So anytime that you want to do a blood glucose, you want to sit there and get it and do it right away. You don't want to let it go sit for an hour on a shelf and then go do it, because you're going to get an abnormally low reading on it. And so hemoglobin itself is made up of a heme pigment portion. It's produced in the mitochondria, and it contains iron. And then you have a globin protein. So you've got a globin protein. So heme is this protein portion that's pigmented that contains iron. Globin is a protein produced by ribosomes. And what you'll find out is with a hemoglobin molecule is you'll have one globin in the middle, and you'll have 4 hemes actually attached to it. And so every heme group can actually carry 1 oxygen. And so there's 4 heme groups attached to each one of those globin molecules, meaning that each molecule of hemoglobin, when you put it together, carries 4 oxygens. And the key to carrying oxygen is the iron within the heme. So basically, one reason why red blood cells look red is that when you put oxygen to them, basically what happens when you put oxygen to iron outside, well, it rusts. Well, basically you're getting a rust color from the interaction of oxygen to the iron. And then when it gets to the cellular level and there's a lower concentration of iron, actually some of the oxygen diffuses back off of the iron. Not all of it, but most of it does. And it releases it to the tissues, and then carbon dioxide and waste are picked back up. Normal hemoglobin types we find in fetuses is what we call hemoglobin E. And so this is the type that's going to be found in embryonic form. And it's a little bit different than the other types of hemoglobins. And then as you mature, you actually go to what they call hemoglobin F, found during the fetal period. And you're just going to be there from mid to late gestation and up to a couple of months after birth. And then you get the normal adult hemoglobin after a couple of months and then throughout the lifespan of the animal. Oxyhemoglobin is hemoglobin carrying oxygen. So 1 oxygen molecule is associated with each iron on the molecule. And then deoxyhemoglobin is that has released its oxygen. Carbon dioxide diffuses somewhat back in the red blood cells and is also carried into the plasma back around to the lungs in its transport. And the thing to know about carbon dioxide is it actually forms something called carbonic acid. So what it does is this right here is water, and carbon dioxide goes together, makes, this is actually carbonic acid right here, and then this carbonic acid will actually turn over into what we know as bicarbonate, which we'll get into later on, which is very important for buffering. And it'll release this hydrogen ion. This usually goes over and to lactic acid to make lactate. So lactic acid really likes to have that hydrogen, so it'll kind of pull it away. But it actually deionizes. The thing is when you get back to the lung, any free hydrogen goes back to this bicarbonate right here, makes carbonic acid again, and then carbonic acid goes back into water, and then the carbon dioxide that you breathe back out. Lifespan of red blood cells. This is going to vary with age. And so one thing to know about this right here is when you're looking at the lifespan, dogs is about 110 days. Cats end up being about 68 days. Horses and sheep is about 150 days. Cows are about 160 days. And a mouse's red blood cells are only 20 to 30 days. One thing that happens is that red blood cells are stored in the spleen, and the spleen is also very important for the fact of this is also where senescence or the aging process takes place, and the spleen is very important for actually removing aged red blood cells. So it acts both as a storage place and then also as a place to actually remove the aging in red blood cells that need to be destroyed, or any damaged red blood cells that need to be destroyed. So there's a decrease in enzyme activity as these red blood cells get older. They lose their formability, so it's much easier to be deformed. They're not as round. Their volume decreases, and they actually get little chips. They get beat up and banged up through the capillaries, and then it gets to a point to where uh, they're no longer being efficient, and so they need to be replaced. So there's actually these macrophages within the spleen, and they're actually looking for red blood cells that are actually kind of leaking their contents, broke on the outside. They recognize these proteins from their plasma membranes, and then they actually take them and they actually phagocytize them and actually break down the red blood cells back into their components. And so certain parts are going to be recycled, such as the iron is going to be recycled and used, some of the proteins and stuff, and other parts are going to be eliminated as wastes. And a lot of times those wastes are going to be eliminated through the bile. Extravascular hemolysis is red blood cells being destroyed. Uh, and then what happens with them is the iron goes back to the bone marrow. A lot of your amino acids from the globulin molecules are actually transported to the liver for reuse and to help in making the bile salts. Heme is converted also into bilirubin, and so bilirubin is bound to albumin and transported to the liver. And so the heme also plays a role in helping to make bile. And actually the heme is the part that actually gives bile kind of its greenish color. And so it's actually conjugated into what they call glucuronic acid, and then it's excreted as a bile pigment into the intestines. It's slightly alkaline, and so it actually helps to combat some of the acidity that you're going to find uh, in the stomach acids when it gets to the small intestine. The other thing is the bacteria in the body will actually convert it to what we call urobilinogen, and some of this is actually reabsorbed and eliminated through the urine as urobilin. And so you'll actually see this when you go to do urinalysis on dipstick tests. And then some of it's going to be excreted in the stool, and we can actually measure some of that. And then some of it's actually brought back in and can actually be reused. So that's extravascular. Intravascular is inside of the blood cells, and so in circulation, sometimes red blood cells will actually lyse, and there's several different reasons for doing this. And the hemoglobin is released directly into the bloodstream. And one of the things that comes with this is actually icterus. So if you actually have red blood cells that are being destroyed in intravascular hemolysis, you actually get a lot of icterus from this. You can with enough from extravascular too in the spleen if all the red blood cells are being destroyed with certain parasites like anaplasmosis. But a lot of times red blood cells, whenever we have them in intravascular hemolysis, you just about always end up seeing some form of icterus that comes along with it. You don't always with extravascular. It has to be something really special, like something, a disease like anaplasmosis to see it. But anytime you have intervascular or intravascular, you will. Things that will cause this is drugs. So giving certain drugs too fast or certain drugs period can actually cause intravascular hemolysis. Another thing is fragmentation and destruction. So if we start having these odd clots formed in the bloodstream, and it's almost like they put a guide wire across the endothelium inside the veins and the arteries, the red blood cells will come past and it'll actually shear them in two. The other thing that will cause this is antibodies. So this is the classic Rh factor that you see in neonates, where mom has one blood type, baby has another blood type, and then mom's immune system actually starts destroying the fetuses or the newborn's blood cells. And so that's another case of intravascular hemolysis that happens. And so all the hemoglobin and all these different factors are released directly into the bloodstream. So unconjugated hemoglobin attaches to haptoglobin. It's transported to the macrophages in the liver, and it's processed, and so the iron is kept and used again. And then the unconjugated hemoglobin, the plasma, is going to be eliminated through the urine. And so one thing that you'll actually see with intravascular hemolysis is we actually end up seeing hemoglobinuria. So what you actually end up seeing is kind of red urine or coffee ground looking or coffee color looking urine. So it can be anywhere from a red to a brown color, and that's a sign that we're having intravascular hemolysis going on. Anemia, this is any decrease in either one of two things. The main thing we measure it by is decrease in red blood cell numbers, and that's the classical definition. We can also have forms of anemia where we don't have enough hemoglobin to carry oxygen. Any which way it goes with anemia, one of the biggest issues that we're going to have with it is decreased oxygen carrying capacity of the blood. So it can be from low numbers of circulating red blood cells, and it can also be from insufficient hemoglobin production. Polycythemia is an increased number of red blood cells. There's relative polycythemia. This is hemoconcentration due to blood loss. So when we see this type of polycythemia, it's usually due to dehydration from things like vomiting or diarrhea. Compensatory polycythemia, this is a result of hypoxia. So people that live at higher altitudes, so if you want to go live up in Pagosa Springs, Colorado at 7,000 to 8,000 feet, you'll find a lot of people up there have polycythemia, and it's actually compensatory. And a lot of the animals do too, because the higher altitude, there's less oxygen up there. So what the body does is it makes more red blood cells to make up for that. And then also some animals in congestive heart failure that are not getting enough circulation, the body will do a compensatory polycythemia. There's also a rare bone marrow disorder called polycythemia rubra vera. The cause is unknown, but these people and these animals will have polycythemia, and it's not relative and it's not compensatory. Most of the time, it's not much of a problem, but sometimes it can become a problem because it can actually, too many red blood cells can actually slow down circulation. That can actually start clogging and causing issues. And so, some animals with polycythemia rubra vera, it's just kind of oh, well, that's an interesting. Others of them you'll actually have clinical illness with. So platelets, these are actually cytoplasmic fragments of bone marrow from megakaryocytes. And their biggest thing that they do is they provide a scaffolding for blood clotting to take place. Making platelets and megakaryocytes is what we call thrombopoiesis, so it's actually the production right there. And so the megakaryocyte goes in incomplete mitosis during maturation, so it doesn't quite go all the way through mitosis. The nuclei divides, but the cytoplasm doesn't, and the resulting multinucleated cell has all this extra cytoplasm going on with it. It's quite large, and it'll actually start breaking off pieces of its cytoplasm, that cell membrane right there, plasma membranes, and they'll pinch them off and they'll send them out into the bloodstream. Circulating platelets are small with numerous small purple granules in them. These granules contain a lot of your different clotting factors and calcium, and when they're activated, they actually release these. And usually, if in the peripheral bloodstream, if the blood vessel is intact, the platelet will not be activated. But once you damage the lining of the endothelium of the blood vessel, it actually sends out chemical cascade that tells the platelets to actually activate and actually start releasing these little granules with clotting factors and calcium, and it literally tries to plug wherever this damage is at. So they maintain vascular integrity, so they actually sit there and keep blood from flowing out anywhere where there's a possible leak. They actually help to release growth factors to actually cause the endothelium and the blood vessel to heal itself. They actually have that clot formation, that plug we've been talking about that keeps bleeding from going on and on. And they play a big role with fibrinogen, making sure that they get scar tissue in there to actually stabilize that hemostatic plug or that blood clot. So it kind of helps to make a mesh around the platelets to where the bleeding doesn't keep continuing. White blood cells we have called leukocytes also. We kind of put them in different classifications. We look, number one, at their functions, secondly, at presence or absence of granules, and lastly, at the nuclear shape. And so we have this big graph up here, and so we have neutrophils, eosinophils, basophils, monocytes known as macrophages, and then lymphocytes, B cells, and T cells. So neutrophils, eosinophils, and basophils, all of these have a polymorphonuclear shape, meaning that there's multiple, they're multiply nucleated. So they have multiple, not nucleuses, but their nucleuses are actually segmented and they look like they have multiple nucleuses. They're also all granulated. So that's a really big thing, is that you can actually look at them and there's actual little granules and stains. Eosinophils have a stain that can be anywhere from orange to red. Basophils stain blue. Neutrophils don't stain that heavily. You can pick up a little bit of stain in there, but you can make out more of the outlines. They're not quite as bright and as basophils and eosinophils are. Some of their different functions, neutrophils and these granulocytes, they're going to be important for phagocytosis, so actually getting in there and taking care of bacteria or funguses or any type of foreign invaders or dead cells and the organelles of dead cells. And their actions are throughout body tissues. Eosinophils, they're very important in allergic reactions and anaphylaxis, and also they can do phagocytosis. They're also can react to, a lot of times, actually to parasites. They can react to bacteria too, but they're very important in actually trying to take down larval migrants. And so they're going to be found throughout the body. Basophils, their main thing seems to be initiation of immune response. So they call in things like neutrophils and eosinophils to, hey, there's something challenging the immune system, come take care of it. And the other thing is they play a very major role in allergic reactions and anaphylaxis. And so all these live out in the body tissue. You find some in the bloodstream, but the vast majority of these cells end up leaving the bloodstream and actually hanging out in the tissues. Monocytes, also known as the macrophage, they don't have any granules. They're called pleomorphic, so they're not segmented, but when you look at the nucleus, it's round. And we'll go back a couple slides here, and look at the shape. So, the monocyte, it's kind of a horseshoe shape. You can see there's little lobules of it going all the way around. You can see here where these have got segmented nucleuses, so it's one nucleus, but they're all in different segments. You can see how the basophil and eosinophils have this orange stain in the eosinophils, the blue ones in the basophils. You can see this is granule and a neutrophil, but they're just not nearly as stand out as much as your basophils and eosinophils. And when we get to our lymphocytes, you'll see right here that they're mononuclear. And so you can see it's pleomorphic where it's horseshoe shaped. You can see there's little lobules, but it's not fully segmented like the polymorphonuclear cells are. Biggest job is it does phagocytosis, bacteria, and also other cells. And so it's very important for that process of taking care of bacteria, taking care of funguses, viruses, and then also any cells that were destroyed or in the organelles. It cleans up all that. And it's found both in the blood and then really in large amounts in body tissues. The other thing it does is it looks for antigens. And so a lot. So it actually processes and looks for a lot of these signs of infections. Finally, we get to our mononuclear cells, and they're very important for immunity. We have our B lymphocytes, which stands for bursal factor. These actually live in your lymph nodes, and their primary job is antibody production. Something comes along that's foreign to the body, it's brought to the attention of the B cell. The B cell looks at it, figures out what's the little projections off of the bacteria or off of the virus, and it actually codes and makes antibodies to match up with that would actually attach to that virus or that bacteria or whatever that foreign substance is. T lymphocytes are what's known as wandering cells, and therefore the thymus, which is actually located in the mediastinum. They do some maturation here. They come back here to the thymus from time to time, and they make cytokine production, which is an immune media, causes immune mediation, and they are part of cell-mediated immunity. So they actually go and wander out in the tissue, and they actually find things that look foreign. They make these cytokines, and this cytokine kind of goes on and triggers to other cells that there's something wrong, white blood cells, that there's something here that needs to be taken care of. The other thing they can do is they can actually pass that information on to B cells, and so B cells then can actually take whatever comes by with that cytokine, and then it can actually sit there, grab it, and process it and say, hey, this is something abnormal, so let me make an antibody for it. And so these are found in the lymphoid tissue, but they're also found throughout the tissue in the body. One thing they don't have on this chart that's another type of lymphocyte, and we're just learning more about it, but it plays an important role in actually regulating cells in the body and looking for cancer cells, is the natural killer cells. And they're actually a lymphocyte. They wander around like a T cell, but they go looking for cancer cells, and they're the ones that are responsible for taking out cancer cells. And so a lot of our cancer research now is actually seeing how do we get these natural killer cells to try to take care of cancer before it gets out of hand, or how can we use them to help us fight cancer? How can we produce their production and get them to go and attack those cancer cells? Leukopoiesis occurs in the red bone marrow, so this is where they're all going to be made at, like everything else. And then some lymphocytes further mature outside of the bone marrow, mostly your T cells. Your T cells go to the thymus. That's where they do their final maturation at. Same pluripotent stem cells for red blood cells and megakaryocytes. And each type of white blood cells has its own stimulus for production, its own hormone that says, hey, let's make this. So granulopoiesis is going to be making neutrophils, eosinophils, and basophils. And these are based on the need of the body, what the body's needing at that time. Initially, when you look at them, there's no cytoplasmic granules, and then the granules are non-specific granules when they're formed. And later on in maturation, they're going to make their more specific granules. Granules contain different substances depending on their function. So like neutrophils, granules actually have a lot of lysosomes, enzymes, to use to break down things like viruses and funguses and bacteria that happen to invade the body when they engulf them through phagocytosis. They pretty much dissolve anything that gets on the inside. So when you sit here and you look at them, you can see this is what the nucleus looks like. It's multi-segmented. It makes it look like it's multinuclear, but it's not. Part of the reason why we believe it's so segmented like this is they're actually very metabolically active, unbelievably metabolically active, these segmented white blood cells are, and neutrophils are. They're the most numerous white blood cell in circulation in the dog, horse, and cat. Ruminants are a little bit different. They have more lymphocytes. But overall, it doesn't matter which species that you look at. When you get away from circulation and everything else, in all species across the board, there's more neutrophils than anything else. In ruminants, there just seems to be, they don't stay in the bloodstream as much. They seem to go out in the tissues a lot more, so you don't see as much. But if you were to take and count all the ones in the tissue, definitely there's more neutrophils in all species. But when we actually look at a blood count in dogs, horses, and cats, neutrophils are by far the leading white blood cells that you'll look at. Instead, the granules don't stain nearly as good as we saw with the eosinophil or the basophil. They mature in some mature in circulation, and 2 to 5 nuclear segments are joined together by a stream of chromatin. Immature neutrophils have a horseshoe-shaped band. They almost look like a monocyte, except they're smaller, and they're what they call band neutrophils. Most of the time, the ones that you're going to see in circulation are going to be your mature ones. Usually, we don't see that many band neutrophils. You'll see one occasionally in dogs and cats. You should never see them in horses in circulation. But you'll see occasionally one or two, but most of the time they're going to stay in the bone marrow until they're fully matured and fully segmented. Instead, the main thing they're going to do is phagocytosis, destroying bacteria, viruses, other invaders that come into the body. One of the things that they actually do is extravasation, where they actually leave the bloodstream and they actually go between the endothelial cells, that little bit of uh, epithelium that lines the inside of the blood vessels, and they can actually do something called diapedesis, where they actually flatten themselves out and squeeze right in between the junctions here between these epithelial cells and go out in the tissue. And usually where they go to is something called chemotaxis. They're actually looking for certain chemicals, and when those chemicals they see that are a sign of inflammation or a sign that something's going on, they actually scoot their way out of the blood vessel, and then they actually go right on into that area where they're needed. The other thing why there's a lot of them that actually happen to cling on to the walls of the that don't free float, but they actually kind of roll and cling on to the walls of the blood vessels. And that's the other thing in ruminants is they seem to cling a lot more to the side of the epithelium right here on the endothelium. And so you don't actually get to draw as much of those out because they seem to cling a bit stronger right there. But when you get chemotaxis, they leave from clinging on or rolling along here, and they go off into tissues. Their action, outer membrane takes in microorganisms. They have lysosomes that surround the microorganisms, and then those lysosomes engulf those microorganisms and dissolves them, and then they remove them out through exocytosis as waste after they've been destroyed and they're no longer causing any chance of any infection or inflammation. They also have hydrogen peroxide inside of neutrophils, which is very important for cells that are anaerobic. And so when this hydrogen peroxide is released, it kills any anaerobic bacteria. And there's also myeloperoxidase released from the neutrophil granules, which actually happens to enhance the bactericidal effects of the hydrogen peroxide. The other thing it can do is it can actually destroy cell walls of microorganisms. So if you remember, bacteria have cell walls around them. So this hydrogen peroxide with the myeloperoxidase actually will sit there and eat and chew up and destroy cell walls of bacteria. Neutrophil counts relatively stable in peripheral blood. And there's a few things that can cause it to go up and down, such as stress. One thing that stress will do in the presence of cortisol is all those neutrophils that were hanging onto the endothelium will actually lose their ability to grip and they just free flow through the blood. So it'll make those numbers go up. If you have something like an infection, that can make it go up. But acute infections or really severe abscesses will actually make it go down because all the neutrophils will go in and concentrate in that one location where they're needed. There is actually a pool where they're stored in the red bone marrow until they're needed for release. And like I said, a lot of them escape out in the tissue. Once they get out in the tissue, they don't actually come back into the circulation again. So you have the circulating pool and then the marginal pool, and this is what I was talking about. There's a lot of them that actually sit there. They grasp onto the side of the small blood vessels. They don't come out off very easily. And then there's actually a storage of them in the spleen. There's a lot of them in the lungs and a lot of them in the GI tract, and they hang out right there in what we call the marginal pool. It's kind of like a reserve area or a resting area for them. And they know they're going to be needed in those areas because these are areas that, especially when you're looking at the lungs and the GI tract and the reproductive tract, that it's exposed to bacteria and somewhat to the outside environment. So there's a lot of invaders in there. And so they kind of wait in this area until they're needed. And then if there's any type of invasion, they get the signal, they go to that area and they take care of that bacteria or whatever foreign is invaded, fungus, virus. Eosinophils, red granules in the cytoplasm. Horses have the most eosinophils. And so you're most likely to see eosinophils in horse blood, but you will occasionally catch them in dog and cow and other ant species too. Percentage of the total white blood counts 0 to 5%, and heavy concentration on the 0. There's a lot of them out in the tissue, but you don't always see them in circulation. Once again, from the bone marrow. And they have the segmented nucleuses, usually 2 lobes, occasionally 3, but usually just 2. Dogs have these round granules with varying size. They're slightly pale staining. They're larger than a neutrophil is, but you can kind of see them with these very large round granules in there, and small granules kind of mixed. And so they don't stain. When I say pale staining, it's a very pale red, not quite as brilliant as what we saw earlier. Cats are numerous small staining ones, and they're rod shaped. So that's how you can tell one from a cat, is that they're rod shaped. Horses have very large, round, beautiful orange granules, stain intensely. And then in pigs, cattle, and sheep, they're pink to red, and they're small, round granules. And these cells are definitely larger than what you're going to see with a neutrophil. There's a bone marrow reserve. There's some in circulation, but most of them stay in the marginal pool, and they migrate through the tissues all the time. Usually when they're being released from the bone marrow within a short period of time, they're going to sit there and actually get in to the tissues, and that's where they're going to want to stay at. You find a lot of them around the GI system or around the respiratory system. They're important for allergies, but they're also very important for taking care of parasites. They have anti-inflammatory granules containing anti-inflammatory substances. They can actually sit there and ingest multiple different types of substances and in immune response, so they can actually do phagocytosis. And they can also release a lot of their granules, and they can have a bactericidal effect to them. But the other thing they do is they can play a role in releasing histamine, calling in more and more white blood cells, such as neutrophils, to come take care of what the problem is. One of the big things that they do is actually taking care of things like protozoas and parasites, and they do that more than they do the phagocytosis and the bactericidal functions. Increasing causes for increases in eosinophils usually is going to be some type of a release, usually in response to either some type of allergic condition or to parasite loads. And usually it's a longer time in the peripheral blood before they're entering the tissues. Another reason for eosinophilia, when we get too many of them. So we've produced a lot of them, and they're just not moving out. But once again, usually that's going to be in response to parasites or to some type of an allergy. Decreased numbers, it's really hard to detect because it's that 0 to 5% that they have. Basophils, the blue granules staining of the cytoplasm, not always visible whenever you do a stain. And sometimes they may completely fill the cytoplasm with all those granules and look totally blue. Least often seen white blood cell in circulation. Dogs have fewer granules than other domestic species. The nucleus usually has 2 or 3 lobes. And usually you're looking at percentage-wise somewhere between 0 to maybe 2 or 3%, they'll say. More chance of seeing these basophils inside of, once again, horses or cattle than you actually do in dogs and cats. One thing is, and this is not a theory, but you'll hear of mast cell tumors, and we now know that mast cell tumors, a mast cell and a basophil is the same thing. A lot of your basophils live out in the tissues, and sometimes they actually become cancerous, and they actually start to differentiate and start to form tumors. And so you'll actually have these masses that come up on the skin. They grow relatively fast, so we take them off. We say they're mast cell tumors. The problems with mast cell tumors is the fact that they're very aggressive tumors. And so usually if you don't get to them fairly quick, then they metastasize throughout the body in a very, very fast way. Some of the things about basophils is they're really important in allergic responses. They release a lot of histamine, a lot more than eosinophils do some, but basophils really do. And this histamine really calls in your neutrophils, other eosinophils, basophils, to work on whatever the problem is. The other thing is they release heparin. So they naturally make heparin, and this is an anticoagulant. And so basophils, the histamine is very important for initiating inflammatory reactions. And the heparin actually helps to keep blood from clotting. And this is important to actually help to get more white blood cells into a location. And a lot of times they're kind of the cells that are important, like when we get a local injury and you get that swelling at that injury and that redness, a lot of times that heparin helps to increase blood flow, which brings in the heat and the redness to the area, and a lot of the soreness comes from all this histamine released from the basophils. Basophilia can be associated with allergic or hypersensitivity reactions, so any type of allergic or hypersensitivity of the body. You'll see a lot of basophils. Basopenia, very difficult to evaluate because it's not uncommon. You can look at a whole blood smear in a dog and a cat and never see a basophil. You can look at a blood smear in a horse or a cow and whole blood smear and maybe come up with one or two. Monocytes, about 5 to 6% of the circulating white blood cells. Uh, they're the largest of all white blood cells. You can tell that they have that horseshoe-shaped uh, nucleus to them, and it's pleomorphic. It's not in segmented, but it's kind of got that unusual shape and kind of got little lobes to it, but it's not segmented. Abundant, plenty of clear staining cytoplasm that's kind of a grayish color. They say grayish blue. They might have vacuoles, but within them, but of varying size, but they don't have granules. So you can see the little vacuoles where they're storing enzymes, but they don't actually have granules. Very important in phagocytosis. You find a lot of these macrophages in places like the spleen and different organs like the liver. They're there to remove any type of foreign invaders from the bloodstream. They also become very important when you have abscesses or ongoing damage or cellular debris going on to a site. So if you have, say, a bruise or something, they play a very big role in cleaning up the cellular debris. They play a very large role in abscesses of actually trying to get rid of the bacteria. So if anything's long going or long standing and ongoing as far as inflammation in the body, you're going to find a lot of monocytes in that area. You have tissue macrophages, and then you have the monocytes that are known as the mononuclear phagocyte system. And so you have the ones that actually live in the tissues, and then you also have the ones that are part of the MPS. Monocytes take care of cellular debris after inflammation. They clean up things from infections. So they're very good at being at phagocytizing things. And they react to a lot of different antigens, and so they're more antigenic. And so presence of a lot of the different antibodies and stuff from the lymphocytes in the immune system triggers a lot of your monocyte and macrophage activity and helps them to ingest foreign invaders. They follow neutrophils from chemotaxis, so your neutrophils are usually your first line of defense, and then the monocytes come and back them up. And they remain at the site of damage or abscesses longer than any other cells. They live a lot longer than neutrophils. They're more hardy. And they're found in circulating blood to phagocytize damaged or dead red blood cells in the spleen. Usually that's where you're going to find them, and in the liver. And then they also pick up any type of microorganisms that happen to get in the blood and get filtered through the spleen and the liver. They'll actually reach out there and grab those. Monocytosis, increased number of monocytes in the blood cell. Usually we see this with chronic inflammatory conditions. So conditions that have been long-standing of infection or inflammation, you have an increased number of monocytes. Decreased number usually is hard to evaluate because they are found in a very low number. They up to 6% of them in the blood. So when you get a low number, it's kind of hard to figure out, is that actually clinically significant. Lymphocytes and the primary circulating white blood cells that we see in ruminants and pigs. They don't have any phagocytic capabilities, so they don't actually engulf and dissolve anything. They either reside in lymphoid tissue or they circulate between lymphoid tissue and the blood. And so there's a couple of different types, and it's which one does which. Come from the same type of bone marrow, depending on what the body's needing is depending on when it makes lymphocytes. So just like, if it's needing more basophils, it'll do it at that time. If it's needing more lymphocytes, it'll make more lymphocytes. Lymphocytes also have an ability to actually clone themselves, and we're actually going to get into that when we start talking about cellular immunity. So types of lymphocytes. You'll find that every lymphocyte actually has surface markers, and they have a differentiated subset for each type of lymphocyte. And so these markers are actually on their surface, and it actually helps to determine which one of these three they're going to be. You have T lymphocyte, which is from the thymus. These are the ones that wander around. They give cellular immunity. They actually go out to the cell, they release those cytokines, they say, hey, this is something that needs to be taken care of by the immune system, by the B cells, antibodies need to be made. And then certain numbers of them actually go back into the thymus. And what they do there is some maturation stuff, but some of the stuff we're not fully sure what they do. B lymphocyte stands for bursa. They're the ones that you're going to find in the highest concentration inside your lymph nodes. So when they're found in circulation, it's because they're going to the lymph nodes. And then you have the natural killer cells, and so these are the ones that go after the body when things are going wrong, like cancer. And so that's one of the big things with natural killer cells that we found out. We found out that there's some antimicrobial activity, but it seems to be more of an immunity against cells that aren't working right in the body. And so there's a lot of research going on, especially with cancers right now, with these natural killer cells. T lymphocytes that go through the thymus before actually going um, out into the bloodstream. So we have what they call pre-T cells, and they're called thymocytes there. And what they're doing is they're maturing and getting ready for their job, and their responsibility is for cell-mediated immunity. They're going to go around, and it's like duck, duck, goose. They go around and they feel everything, and they go duck, 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 and all once they touch bacteria, they put cytokines on it, and they say goose. And of course, that bacteria goes on, it's going to be picked up in the lymphatic system. It's going to be taken to where there's a bunch of B cells, and those B cells are going to read those cytokines, and then they're actually going to make antibodies for it. Most of the lymphocytes that you find in peripheral blood end up being T cells. B cells are the ones that, like I said, they can be found in the bone marrow, but most of them are going to be in your lymph nodes. And the bursa equivalent comes from actually where they're found in a specific organ in birds called the bursa fabricus. Inactive B cells travel through the bloodstream, and then once they get to the lymph node, the spleen, or other lymphoid structures, they activate and they start doing their job. So with the ones you find in circulation, most of the ones in circulation end up being T cells. Occasionally you'll have B cells. You can't look at the difference between a T and a B cells, but the B cells you find have been made in bone marrow, and they're traveling somewhere to lymphatic tissue. B cells are very important for making antibodies. And so what they'll do is they'll actually feel a different antigen, and they'll actually feel the epitope markers on them. And based on those epitope markers, they're actually going to make antibodies that actually match up with these epitope markers. And so these epitope markers are actually unusual things on the surface of bacteria, viruses, and funguses. And so they actually make chemical bacteria that actually will match up with these. And when they match up with these antibodies can do a couple of things. They can actually destroy the microorganism that's invaded, or they can mark it for neutrophils or monocytes to sit there and actually phagocytize and destroy those marked cells that the antibiotics have actually attached to. B lymphocytes give what we call humoral immunity, and so they produce all the antibodies that are going to be circulating throughout your bloodstream. They can actually activate themselves by mitosis and actually make more of themselves, make more of these cells to actually produce more antibodies, more immunoglobulins. So they can produce themselves by normal hemopoiesis in red blood cells, but we actually have these memory cells. If they get triggered inside of the lymph nodes, they actually do mitosis there, and they actually start producing more of themselves to make more of the antibodies against whatever the foreign invader is. So an example of this is we vaccinate a dog for parvovirus. So when we do the vaccination, T cells are going to touch that vaccine. They're going to mark it. They're going to say, this is something foreign. It's going to go to the B cells. The B cell says, oh, what's this? Okay, I'm going to make an antibody against it. You give a booster after the first vaccine, and all at once, the T cell says, this thing's back, marks it, goes to the B cells, and the B cell says, oh my God, it's back. We're getting invaded again. We have to make more antibodies. And so it actually clones or does mitosis on itself. and it makes it to where it can increase the antibody production, and so that's how you get these circulating levels of effective antibodies. And then when the real thing comes along, there's enough antibodies made from this cellular immunity, from this mitosis, the splitting of these B cells, to where you have enough of these antibodies to actually mark and fight the virus when they're actually introduced to it. And so one of the things here with cell memory is, depending on the viruses, is how long the cell memory is going to be. Something toxins like tetanus, this can actually be a lifetime, nearly a lifetime immunity for some animals that they can get from just a few vaccines, including ourselves. We know right now that a tetanus vaccine lasts at least 20 years. There's proof in horses that, if you've done the first round of tetanus and you booster it, there seems to be 10-year immunity. We do it every year because the label tells us to do it every year for tetanus, but it seems like it might be potentially overkill for how much it's given. So we keep doing it because the label tells us to do it, and we don't want to get in trouble if we didn't do it and we advise somebody not to, and then the horse did get tetanus. We would be liable. As long as we tell them every year to follow the label, then we're not. So that's why we do it. But it really seems like there's a really good, strong defense against that. On the other hand, some of these memory cells don't last as long, and one of the memory cells that doesn't last as long, or memory that doesn't hang around as long, as the influenza virus. So that's why you have to constantly get vaccinated against influenza. Natural killer cells, they're not actually specifically antigen driven. And they actually have the ability to kill tumor cells and other cells infected with viruses. So they go around and looking for cells whose DNA is not working right, and they actually kill those. There also seems to be maybe some antimicrobial capacity with them, but that's still being looked into further. Really big research going on with the natural killer cells about how we can actually use these to help us in fighting cancer in people's body. Lymphocyte characteristics. Circulating lymphocytes are classified either as large or small lymphocytes. There's going to be no granules in there. They're going to have nucleuses that are oval or round and non-segmented. Large lymphocytes have this abundant sky blue kind of cytoplasm. Small lymphocytes just have a little bit of cytoplasm. Depending on which species and how that you're looking at is whether or not you're going to have large lymphocytes or small lymphocytes. And then both T cells and B cells have memory. So T cells can actually become specific, looking for specific antigens, and they can make memory cells. This happens in the thymus, and so they can produce more of those memory cells for things that they're actually being exposed to. And then B cells actually make their own memory cells too for antigens. And B cells are specific. They're going to be specific to a specific antigen. They actually make clones, and they only make one type of antibody against one specific antigen. And that's the way that they work. T cells can be a little bit more versatile than that. One thing is with memory cells is you get this quicker and this large, really fast response to any type of initial immune challenge. If there's already been that memory there, whenever there is a second or third or fourth challenge to the immune system by that pathogen, each time it gets a quicker and stronger response to take care of it. Lymphocytosis, increased number of lymphocytes. Can be cancer like leukemia. Chronic infection is probably the most common reason why we see lymphocytosis. Also, things like epinephrine release can also cause lymphocytosis to occur. There's a lot of different things that can be significant enough to cause this leukocytosis throughout the bloodstream. So it can definitely raise the number of total white blood cells from having lymphocytosis. So this can be very significant. Lymphopenia, decreased number. Things like corticosteroids. So giving something like dexamethasone can definitely decrease these. Any type of immune deficiency diseases, such as FIV, will cause this. And acute viral diseases will actually cause this. So a lot of times we'll see lymphocytes go down in acute viral diseases, probably because the T lymphocytes have actually left the bloodstream and are going out trying to identify and take care of that virus. Lymphatic system. This is going to be actually a system to actually try to bring back into the body a lot of this extracellular fluid or this interstitial fluid and actually try to filtrate it out. And there's a series of vessels and ducts, and so these lymphatic vessels, they look a lot like veins. They're thin-walled, not much smooth muscle to them. They actually have valves to make sure everything moves in the right way, and they carry interstitial fluid, and they actually help to circulate it. And it's amazing at how much the interstitial fluid is actually sat there and circulated. So these lymphatic vessels come from the tissue, they go through lymph nodes, they get filtered, they go through another lymphatic vessel to another lymph node, they get filtered. Sometimes they might be filtered 4 or 5 times through different lymph nodes, sometimes 2 or 3 times. Eventually they make it back to one of the vena cavas near the heart, where this lymphatic fluid actually enters back into the bloodstream, and then is circulated back through the body. And what this does is it actually keeps wastes moving away from the cells, so not all of it diffuses out into the capillaries. And the other thing it does is it actually, with the intestinal tract, it actually helps to bring nutrients such as lipids back into circulation. And then filtering this out, you're also going to get a chance for the liver to take care of toxins from cells, and the kidneys to actually get rid of some of the wastes, along with the lungs getting rid of things like carbon dioxide. When we look at the lymphatic system, this is actually going to be lymph nodes are involved with this, spleen, thymus, tonsils, gut-associated lymph tissue, what we know as GALT. Also, the liver plays a role in the lymphatic system. So there's all these different areas that actually play a role in immunity in the lymphatic system here. So you have these vessels, lymphatic vessels. They do carry some fats and proteins from the GI tract, and those will be deposited back into circulation to where they can go through to the liver. That carries hormones. There's a lot of cells that carry hormones, and they're spread through the lymphatic system. You'll find T cells that circulate from the blood into the lymph nodes and back out into the blood through the lymphatic fluid. And then your B cells are actually found within the lymph tissue, and once they get there in the lymph nodes, they don't go and recirculate. So, it's really not excess interstitial fluid, but this interstitial fluid is constantly, as we move our bodies around, is constantly being pumped and moved. And there's also a pull as the heart beats, actually pulling some of this lymphatic fluid through too. And all it is interstitial fluid around the cells. And actually the body has a way of actually recirculating all the fluid in your interstitial space about every 2 to 3 hours. It's in a normal body, it's actually fully recirculated. And so it's keeping the stuff moving. Well, like I said, it's trying to pick up nutrients, it's trying to pick up hormones, it's also trying to get rid of waste, trying to keep everything from being stagnant. And what happens and how we get this fluid in the interstitial space is after this lymphatic capillary here and this fluid goes up to the heart, it gets circulated. The pressure from the arteries when it gets out into the capillaries actually forces some of this fluid back out into the tissue. And so part of the fluid in the body and your cells, of course your blood cells circulate through the blood vessels, but the fluid, part of it stays in here, but part of it gets forced out into the tissues. And so if there was no way to recover it back through the lymphatic system, what would happen is everything would swell, and eventually it would collapse these vessels and no circulation would happen. So part of this is forced out, and then it's picked up by the lymphatic system, and eventually it's carried back in and around for circulation again. Lymphatic capillaries join together to form larger and larger lymph vessels. They have these one-way valves to keep it from flowing backwards, and it helps to propel that lymph fluid forward towards the heart. And then these lymphatic vessels eventually join this thoracic duct that actually empties into the vena cava just before it enters the heart. And along the way, the lymphatic fluid is going to be actually filtered by lymph nodes and lymphocytes, and you're going to have different macrophages that are going to be actually in those lymph nodes. And if there's any bacteria or viruses, anything like that that's tagged by the antibodies or by those lymphocytes, then those macrophages come in there and they actually phagocytize and destroy whatever has been picked up in that lymph node as abnormal. Lymph fluid itself is transparent to translucent liquid most of the time, so it's kind of this clear looking fluid. There's very little cell. There's a lot of water, sugar, and electrolytes than in plasma. There's also some larger proteins actually found than in plasma. And then you get this chyle, which is this lymph from the digestive tract. And it's almost a whitish appearance. And that's because it's full of lipids and fats. So it's going to circulate that fluid, get rid of waste material. It's also going to sit there and bring up nutrients and stuff for the body. It's going to filtrate that fluid, try to get rid of cellular debris, microorganisms, foreign matters, and then, it's part of a transportation system. When we look at lymph nodes, they're small kidney bean shaped or small kidney shaped structures located along those lymphatic vessels. And they have this connective tissue, thick capsule on the outside. And this capsule has branches called trabeculae that actually extend into the body of the lymph node. We have afferent lymph vessels which carry fluid into the lymph node, and efferent, so E for exit, vessels that actually exit the lymph node. And they actually exit in this indented area called the hilus. So when we look at a lymph node, this is the afferent. So you have multiple lymph ducts that actually come in. And then the fluid comes into this area. There's a bunch of lymph tissues that have B cells and some T cells in this area right here. And then when it comes towards the hilus to these efferent vessels, this is where it crosses the macrophages. And anything that gets tagged out here as foreign or needs to be taken care of or removed, dissolved, engulfed, whatever, when it comes over here towards the hilus and these macrophages, these are the ones that actually do that in this trabeculae area. The outer area over here is called the cortex. So you have the capsule, but this capsule on the outside, but this area out here is the cortex. And then when you get near the hilus, this is going to be the medulla. And it's actually named the same way. When we go to look at the kidney, you'll find that the outer portion of the kidney out here is called the cortex. When you get near the hilus, it's called the medulla. And so it actually contains a lot of the same names that the kidney will. Spleen is the largest single organ in the lymphatic system. There's another one that we'll get to that overall is larger in size, but the spleen is the single largest lymphatic organ. And it's located on the left side of the abdomen, kind of right below your stomach, and it kind of goes across towards the right side, near the rumen and ruminants. And it's, like I said, the largest single. And it's got a very thick, fibrous outer capsule to it. There's a lot of smooth muscle in there. It stores red blood cells, and that smooth muscle can contract and send out a lot of red blood cells when it needs to. But a big thing that it actually does is it has a bunch of trabeculae in there. The blood actually circulates through, and then you've actually got B lymphocytes in there looking for foreign invaders in the blood. And then you have macrophages that are also in there taking care of any of those foreign invaders. The other thing that happens in the spleen is, we had already said, is it's taking care of any damaged red blood cells and platelets. It actually breaks down and phagocytizes those. You have this area out here that they call the sinuses, which are full of the red blood cells, and then you have the inner area of white pulp. This is where all of your lymphoid tissue is, all your lymphocytes and stuff, and then interspersed in there you have your macrophages and this really, really thick capsule on the outside. And you have these blood vessels that come in through the trabeculae, blood circulates through here, and then circulates back out. So blood storage, removal of foreign matter, we already talked about. Removing dead or dying or abnormal red blood cells also plays a role with some of the platelets. And then there is some lymphocyte cloning in the white blood cells. Once again, in that immune response, you have those memory B cells that live in there, that if they see something foreign, that they can start going and developing themselves really quickly to make that immune response and produce those antibodies that are needed. The thymus is located caudal to the neck and in the cranial-thoracic areas and the cranial mediastinum. It's more prominent in young animals and actually plays a role in making white blood cells in younger animals and in fetuses. And then as you mature, the thing it does is it processes thymocytes that end up turning into T cells, and certain T cells come back there, and we're not really sure, but somehow or another they pass off some of their immunity to these thymocytes. Also, when there's going to be cell memory and they clone themselves, they seem to do this more when they come back to the thymus. Tonsils, these are nodules of peripheral lymphoid tissue. They're actually not covered with a capsule, so they're like a lymph node without the outer capsule, and they're going to be found on mucosal surfaces all over the body. One that people know most commonly is the tonsils in the back of your throat and the pharynx. But you also have them in your larynx, your intestinal system, reproductive system, and the prepuce and the vagina. So anywhere where you have mucous membranes at, you're going to actually find these tonsils throughout. And their main job is to actually try to pick up things and stop the spread of infections from the respiratory, the digestive, or the reproductive system. And they actually pick up things. They actually sit there and mark them, put the antibodies on them, and start them into the lymphatic system and drainage system to where they can go along, and whatever those foreign invaders are can actually be destroyed by those macrophages. GALT is gut-associated lymphoid tissue. So along all of your intestinal tract, there's all these lymph nodes in the mucosa and the submucosa. If you were to lump it all together, it would be the largest single organ, but it's not a single organ, it's multiple organ. And so it ends up being the largest lymphoid organ because it's spread out, but it's diffuse. So the spleen is the largest single organ. But if you're going to look at all the lymphoid tissue and you say, well, what is the most lymphoid tissue in the body? It's going to be this gut-associated lymphoid tissue. And it's throughout, from your stomach all the way down to your rectum, is full of this GALT. And it's basically just lymph nodes that are embedded within the mucosa and the submucosa of the intestinal tract. The immune system, its main thing is to actually destroy and get rid of anything that's foreign or trying to harm or destroy the body. And that's its main system, main function of the immune system. And so, lysis of foreign cell membranes and activation of pathogenic organisms or chemical substances, agglutination of cells or molecules or precipitation of them when needed in response to inflammation, phagocytosis and destruction of foreign cells, and also any damaged cells that are dead, they actually go in there and do the same thing with the immune system. You have nonspecific immunity, things like skin, mucous membrane, chemical barriers like hydrochloric acid, inflammatory response of tissue, which provokes the release of things like histamine and different things like interleukins. And then you have phagocytosis by those neutrophils, the monocytes, and those macrophages. Specific immunity, or also non-specific immunity, is your natural killer cells that come into contact with tumors or other cells that have viruses. And then interferon, which is a protein produced in response to viruses, and it inhibits development and spread of other viruses. Complement groups are groups of enzymes in plasma that can be activated by the attachment of an antibody to an antigen. And so we call this fixation, and this actually makes a cascade of reactions that results in the antigen actually being lysed. So if this is a bacteria, all of these antibodies come to it, they attach to this cell wall of the bacteria, they actually eat holes into it, and it actually causes the bacteria to burst. Specific immunity is the antibodies actually linked to the B cells. And so they actually attack one specific antigen. And T cells also can do the same thing. They respond to one specific antigen. So the response is initiated as a reaction to whatever this foreign invader is. And it's actually looking for that marker on that foreign invader's organism cell wall. And then once it actually sees that, then it's going to actually link up those antibodies to it. And those antibodies are actually going to either mark or to try to destroy that actual foreign invader. So when we actually look at different types of what we call humoral or versus cell-mediated immunity here, when we look at humoral response, this is going to be our B cells. Substances produced are going to be our immunoglobulins, and so the B cells and the plasma cells in the lymphoid tissue release the antibodies. Memory cells are produced. Cellular immunity is going to be these T cells, and they actually kind of form themselves into what we call cytotoxic T cells. So these are ones that actually make cytotoxin. Helper T cells, and so these helper T cells actually go and mark a lot of these different foreign invaders. And suppressor T cells after antigenic stimulation. So they kind of suppress a little bit of the immune system. They produce lymphokines, so they don't make immunoglobulins or antibodies. What they do instead is they actually make markers to say, hey, I need an antibody made to this. or they make lymphokines that can actually attach and actually destroy whatever the foreign invader is. And they travel throughout the body. That's the thing, is they're the wandering cell, and they do produce memory cells too. Types of immunoglobulins. You're going to have IgG. This is made during the first exposure to any type of antigen. IgM is made when there's been an exposure to an antigen over a long period of time. IgM is the most common in circulation in the adults. IgG is lower. IgG is more common in neonates. IgA leaves the blood and enters places like the intestinal tract and the respiratory tract, and actually plays a role in providing mucosal surfaces with protection against foreign invaders. So IgA actually lines itself up along the mucous membranes looking for anything foreign coming in. IgE, we know plays a very important role in allergic responses. So if you have an allergy to something, IgE is the one that actually plays a very important role with it in causing allergies. And then we now know there's an IgD, and we're still not quite sure what IgD does. Passive immunity. This is when immunity is actually going to come from antibodies from another animal. And this is usually most often seen with newborns. No newborn's born with its own immune system competent. You have the tools there, you have the cells to build, you have all that, but you don't have the exposure to be able to build those antibodies yet. So you get them from mom. Most of the time in animals, they get it from the first milk called the colostrum, and this is loaded with IgG. Some IgM, but the majority of it's IgG that the mother produces in the mammary gland, and they're able to ingest it in their body. And then it actually hangs around until the neonate starts to mature into adolescence and actually gets a competent immune system, and their immune system is able to make their own antibodies. And this is why vaccinations are so important in young animals, because we're trying to vaccinate in this transition between mom's immunity from this passive immunity to where we have active immunity, and we're trying to actually get that young puppy or that foal or that calf to start picking up its own immunity. With passive immunity, there's no memory cells. So you have to have active immunity to actually have that, where the body's making and doing its own. But with passive immunity, there is no memory cells produced. You just have the antibodies. So active immunity is exposure to the antigen, stimulates the body's own response. The T cells and B cells are produced. And immunization is the way that we actually activate this through vaccines. We can do it with killed vaccines. We can do it with attenuated vaccines that are live but weak. And then we actually have modified live virus vaccines that simulate very closely what the infection would be like without actually causing the infection. So that is Chapter 9 with blood, lymph, and the immune system.